Welcome back. This is now going to be the third part of chapter 23, the digestive system. This is really kind of long, so let's go and start the ball rolling so we can finish this last part on the digestive system. We will start at this end already on the small intestine. We have finished already in parts 1 and 2, and this third part starts with the anatomy and physiology of the small intestine. The small intestine is made up of three segments. You have a very short duodenum, which is about 8 inches long. And then you have a very long jejunum, and then the ileum also, which is at the distal part, distal end. The digestion as well as absorption of nutrients happen extensively along the entire length of the intestine. Then after that, we're going to get into the large intestine and eventually into the all the way into the elimination process of our fecal matter. So let's move it. These are accessory digestive organs only. The liver, the gallbladder, the pancreas are very important, very vital in the process of digestion. Now let's take a look first at the blood supply. How does the absorption of nutrients get into the blood? Well, first of all, before it absorbs anything, it will have to move around and it will need its own nutrients because it has its own muscles. The system, the gut itself has its own muscles, has its own tissues and cells that needs to be nourished and kept alive and kept healthy. And that is through the um, nutrition or coming from the arteries that supplies blood in the gut. So the celiac artery, which is actually like under the diaphragm only, the first branch under the diaphragm, the celiac artery, is the one that supplies blood to the foregut, and the foregut is actually made up of the esophagus, your stomach, as well as your duodenum. Now, any absorption that we have or that happens in the stomach is actually going to be brought the into the vena cava, okay, and then from the vena cava, it is going to be transported via the hepatic portal vein into the liver. So all of the digestive nutrients that has been like absorbed and passed into the bloodstream would have to go first into the liver for processing. And we will discuss later on what the function is of the liver, not necessarily in this chapter. Right here, we're just passing through the what I call this the function of the liver as an accessory organ to the digestion itself. But for the entire function of the digestive system in our nutrition and metabolism, it's going to be a separate chapter. It's going to be on the next chapter that we are going to follow in metabolism and digestion. That's going to be um, besides going over the different things that we need in our nutrition, we will have to discuss all of the metabolic work of the liver, which is really one of the most important digestive organ, or one of the most important organ in the body, and it is also the biggest organ inside our body. All right, then you have also your superior mesenteric artery, which actually supplies blood into the midgut. And when you say the midgut, you're talking about the entire small intestine and about 3/4 of the large intestine. The inferior mesenteric artery supplies blood into the end gut, which is actually the descending colon and the sigmoid colon and the anus. Now, all of this are going to be absorbed by its venous counterpart, and will now be whatever it is that they absorb, water and all of the nutrients will now be transported into the liver for processing via the hepatic portal system. And then after they have been processed, which means, well, um, pretty much to summarize the processes inside the liver, all of the macromolecules that we have eaten coming from the food have um, that we have actually eaten, all of the macromolecules that we have digested, pretty much, let's say if we eat a lot of chicken breast, and this is our favorite chicken breast. That doesn't mean that our muscles will turn into chicken breast. No, we digest them into smallest particle, which is actually absorbable, the amino acid components, and then in the liver, these amino acid components are now going to be built back into proteins, but not chicken breast proteins. It's going to be this time human proteins because now it's going to be part of our body. And it all happens right here in the liver. And then once it is metabolically prepared, it's now going to be transported through the vena cava into the different organs and cells so that these organs and cells can use this in our own this particular proteins in our own metabolism. So that is only in a nutshell. So here, what is actually showing here are structural modifications of the small intestine that will increase its surface area for digestion and absorption. See, the small intestine, like your stomach is actually drawn into folds, which are called ruga. And the small intestine is also drawn into folds. In the duodenum of the small intestine, you have circular folds. It's thrown into circular folds, which are called the plica circularis. And these plicae are like synonymous with the villi of, although the plica circularis is actually like visible to our naked eyes, unlike the villi, which is actually very microscopic. The the microvilli of the small intestine is also microscopic, but they do have some foldings like the ruga, which can be seen with the naked eye. And these modifications provide huge surface area for nutrient absorption. The surface area is increased about 600 times to about 200 meters squares, which is actually the size. Imagine, would you believe it is actually the size of a tennis court? And so these modifications, which is actually just like I mentioned, the circular folds or the plica circularis, can actually force the chyme to slowly spiral through the lumen, allowing more time for nutrient absorption. Meanwhile, there's actually the smaller folds, which is the villi, which is microscopic. These are finger-like projections, which is about 1 millimeter high, with a core that contains very dense capillary bed and lymphatic capillaries called the lacteal for absorption. And the lacteal, just like what I mentioned, and you have to remember this, is actually lymphatic in nature. The microvilli are cytoplasmic extensions of the mucosal cells. It gives a fuzzy appearance that is called the brush border that contains the membrane-bound enzymes, the brush border enzymes, and these enzymes are used for the final carbohydrate and protein digestion. Now, let me see. I think I have in the next slide a nice picture of the brush border uh, itself. Let me see. No, not yet. So here's actually the histology of, just to continue on with the previous one. Oh, I do have a good picture of the microvilli. There you go. That microvilli looks like a brush. That's why they call it brush border. And this cell produces the brush border enzyme, and it's released right through that area there. And therefore, this particular thick area, brushy area, is actually filled with digestive enzyme that is necessary for the final digestion of the macromolecules into smaller, even smaller absorbable units. Now, uh, this is only a sample of columnar cells. That is actually the majority of the cell that lines the intestines. And the intestinal crypts are holes. There you see that. These are tubular glands that are created like this. This is after that. That are created and it's lined to hold the juice or to hold the intestinal juice that is actually secreted within the brush border enzyme. So this is actually where the intestinal juice would be found. Uh, and um, there are 5 types of cells that you find along the uh, brush border, or rather along the intestinal crypts. First, you have the enterocytes, which makes up the bulk of the epithelium. These enterocytes are simple columnar absorptive cells, and they are bound by tight junctions and contain many, many microvilli. And you could see an example here, and you have lots of microvilli on top. The function of which is actually, the function is actually it makes up the villi and absorb nutrients and electrolytes. Now, the crypt's intestinal juice are actually secreted. It's a watery mixture of mucus that acts as carrier fluid for chyme. And they are the ones that are being secreted by the enterocytes. Goblet cells, you will have lots of scattered goblet cells around these villi. These are mucus secreting cells found in the epithelia of the villi and the crypts. The enteroendocrine cells, and the enteroendocrine cells are right here, and these are the green spots right there. Those are enteroendocrine cells. They are the source of enterogastrones. Examples of this is your CCK and secretin. They are found scattered in the villi, but there are some that are in the crypt itself. The Paneth cell. The Paneth cell, that's your Paneth cell right there at the bottom. They are found deep in the crypts, and these are specialized secretory cells that fortify the small intestine's defenses. They are scattered, but because they act as our defense along the intestinal area, they secrete antimicrobial agents such as defensin and lysozyme that can destroy bacteria. You find stem cells. Stem cells are the baby cells that develop into the mature, what you call functioning cells, specialized cells of the small intestine. You find lots of stem cells that are along the walls of the intestine that would continuously divide to produce other cell types. You also find MALT, or the mucosa associated lymphoid tissue that protects the intestine against microorganisms. And you find a lot of mucosa associated lymphoid tissue, most especially along the end, uh, located at the end of the um, lamina propria of the end part of the small intestine, which is the ileum. The ileum is the one that holds more of the Peyer's patches, a lot more than you will find some segregations of small segregations of Peyer's patches in jejunum, but sometimes it's rare, right? But all of the Peyer's patches are found in the ileum. None of the Peyer's patches are found in the duodenum. Although the villi of the duodenum may hold and the jejunum may hold uh, lots of Peyer's patches in them in the intestinal crypts. So They are found, rather, the Peyer's patches are found in the distal end of the small intestine, which is in the ileum, because this is actually where the bacterial numbers would increase because they have been like inside our gut for a longer period of time because they're already at the tail end of digestion. Remember how long food stays in our digestive, in our small intestine, an extra or an additional 2 hours. They stay there, and they stay there longer if you have a lot of fat and proteins and if you have ate a lot. But on the average, they stay there for an extra 2 hours besides the time that they spend in the stomach. They need to stay an additional 2 hours right there in the small intestine to do more of the digestion in the small intestine and the absorption also in the small intestine. And so the lamina propria of the uh, ileum also secrete plasma cells, or also have large number of plasma cells rather, that secrete IgA, or the immunoglobulin A. And its submucosa is made up of areolar connective tissue. Now in the duodenum, let's go back to the duodenum. The duodenal glands of the duodenum secrete alkaline mucus to neutralize the acidic chyme. The villus epithelium would renew itself every 2 to 4 days um, the entire intestinal within the entire three segments of the intestine. Here is a good picture, okay, um, of the villi itself. So this has been enlarged tremendously. You could see this is only of the villi. You could see the mucus granules that are secreted along the brush border, and these are already the brush border enzymes. These are the microvilli forming the brush border. And right here, this line here is the apical membrane, and below that is already the body of the enterocyte. So let's take a look at some homeostatic imbalance of the small intestine. Uh, this homeostatic imbalance that I've mentioned, I just feel that it's actually like something that we need to mention in the study of the digestive system, is the imbalance that is brought about by the use of chemotherapy to treat cancer. And pretty much, we have here a long list of chemotherapeutic um, chemicals, but pretty much the chemotherapy targets rapidly dividing cells, such as the cancer cells. And that's why we use chemotherapy, because these are targeting cancer cells. But inherently or innately, we also normally have our own natural rapidly dividing cells, just like cancer cells. Our epithelium is actually made up of rapidly dividing epithelial cells. And therefore, when we take in chemotherapy to kill the cancer cells, we are also eventually harming and killing a lot of our epithelial cells. And so our intestinal mucosa is lined with pure epithelium. And when you are under chemotherapy, you will feel you won't feel good. You will lose your appetite. You will feel anorexic. You have pain in the stomach. Sometimes you have diarrhea. You have weight loss. There you have systemic infection, most especially in the gut. And sometimes all of these infections could lead to death. So how is that going to help us? You're trying to eliminate cancer so you won't die, but then the chemotherapy may also lead to death. And I say may. Doctors really know very well how much they should actually be putting the patient into chemotherapy in order to avoid uh, death, or to avoid in um, much of the side effects. So one of the things that is actually just very recently been adapted is actually the use of probiotics. Probiotics are mixtures of useful bacteria, which is actually part of our own bacteria already. So the bacteria, when the good bacteria is actually like when the good bacteria would die because of the chemotherapy also, and the antibiotics that we are actually using, the uh, the bad bacteria would be opportunistic, and it will pretty much like cause havoc. So what is the best, one of the best way to help people who are undergoing chemotherapy will be to give them probiotics. These are the these are good bacteria, and by giving them like a boost of good bacteria, then these good bacteria can help in eliminating the effects of the bad bacteria. So let's take a look at the intestinal juice. The intestinal juice is secreted into the duodenum, and it comes from the pancreas. As a matter of fact, most of it is pancreatic juice. And there's actually like 1 to 2 liters secreted daily in response to distension or even irritation of the mucosa. Major stimulus for production is actually hypertonic or the acidic kind. So pretty much when the acidic kind lands, the first drop of acidic kind that lands into the duodenum, immediately the duodenum will send the enteroendocrine cells of the duodenum will send to release the enzymes that would stimulate the production of the pancreatic juice. Depending on the nature of the chyme, if it's heavy in protein and heavy in fat, heavy, if it's loaded with fat and protein, then you know that the pancreatic juice is very much like going to be producing a lot of the lipase and enzymes that will be digesting fats and oil. So the secretion is slightly alkaline and isotonic with the blood plasma. It consists largely of water, but it also contains mucus. And mucus is secreted, additional mucus is secreted by the duodenal glands and the goblet cells of the mucosa, the cells that line the duodenum. The duodenal cells also has another name. They call them the Brunner's gland. Brunner's gland secretes alkaline mucus to um, make sure that the acidic chyme is actually um, mixed. This acidic chyme is going to be mixed with this alkaline thing, and the pH would go to would actually like go up higher than rather than being too acidic. These are peristalsis and segmentation is actually the movement that is happening in the small intestine. Peristalsis is actually the movement or segments or adjacent segments of the alimentary tract organs would alternately contract and relax. And as they are alternately contracting and relaxing, the food is moved distally along the tract and primarily that to propel them and also to do some more mixing to propel them distally going towards the distal end. The other one is actually segmentation. Segmentation is much like, well, this doesn't have, this does not really work on or between adjacent segments. This is non-adjacent segment. So there could be segmentation here and segmentation here. There could be less of the segmentation here. They are not really segmental, but the food is actually moved in segments independent of each other. The movement, though, is different. The movement instead of pushing the food distally like in peristalsis, the movement here is actually churning. So whatever food is actually inside each segment is actually churned in. And the reason for this is I can only think of the analogy of a in a when you are using your dryer. When you're using the dryer, you put all of your wet clothes inside your dryer. The dryer is, once you turn on the dryer will start to blow in hot air. Now, for all of the heavy clothes to be dried, those heavy clothes needs to be fluffed and needs to be mixed, and therefore they need to be thrown around inside the dryer so that every single one of them will receive a dose of the um, of the hot air. The same thing is true with a segment that is undergoing segmentation in the small intestine. The segment that is undergoing segmentation will be going around and around in a circle because the walls of the small intestine, just like what I mentioned, that would have the brush border enzyme. The brush border enzyme is not spurted into the middle of the lumen, just like the pancreatic juice is actually like released into the lumen of the duodenum. The brush border enzymes stay on the border. It stays all along the wall. So you need some of the food to touch the wall and then eventually go to the middle and allow the rest of the food to touch the wall and go to the middle. So they are actually moving the food in a circular manner. That is actually segmentation, and it happens in segments so that every single part will have their own dose of the brush border enzyme. Now, the intensity of these movements is altered by long and short reflexes and hormones. The parasympathetic nervous system would increase motility in the gut, most especially if we just ate food. However, sympathetic innervation would decrease it. That's why we don't have any appetite to eat really if we are very emotional, or if we are sad, or if we are very angry. We lose our appetite to eat because then all of our blood does not pool in the stomach. It's actually pooling somewhere else in order to help us in our fight, flight, fright response. Now, between meals, whenever we have just digested food and before we even eat another meal, our peristalsis would increase. It is initiated by a rise in hormone motilin in the late intestinal phase. It's about every 90 to 120 minutes. Each wave starts distal to a previous wave, referring to as migrating motor complex or the MMC. Meal remnants such as bacteria and debris are moved towards the large intestine through this particular process. The complete trip from the duodenum to the ileum takes approximately 2 hours. That's actually on the average. Now let's take a look at the distal end of your intestine, which is the ileum. And now your undigested particles in the ileum is now ready or getting ready to get into the large intestine. The first stop into the large intestine will be a sac, which you call the cecum. And before the food gets into the sac, it will have to enter through a valve, a gate that will open, which you call the ileocecal valve. The ileocecal valve controls and pretty much the movement of food from the ileum into the cecum. It will only allow, it is actually a unidirectional valve. It allows only food from to travel from the ileum to the cecum and not vice versa. It prevents the food from getting back to the ileum. So the gastroileal reflex, we call it gastroileal reflex, would enhance the force of segmentation in the ileum. gastrin, which is a hormone secreted up like in the proximal end of the intestine and into the when food is on the proximal end of the intestine and into in the stomach, would increase also the motility of the ileum. The ileocecal valve flaps would close when a chyme exerts backward pressure and prevents regurgitation back to the ileum. Now here is actually a table that you can actually use to review and control of the small intestine. This particular table discusses the stimulus and the production of the substance that is involved in digestion, the effects of this substance on the small intestine motility, and the mechanism by which the substance is actually doing while doing its work in moving substances in along the gut itself. And here is another overview, but this is on the function of the gastrointestinal organs. You have here the different organs. You have the mouth, the associated accessories, the pharynx, the esophagus, the stomach. Their major function, which we have already discussed, but this is actually giving you a good view of it. And then there are some additional comments on the functions itself. And here's a continuation from the stomach into the small intestine and then finally the large intestine. Again, you have the function and some comments on the function. So now let's take a look at the large intestine. The large intestine has three unique features that is not seen anywhere else in the gut. First of all, you can see that the large intestine has this band of muscle. This is a band of muscle, which you call the Taenia coli. The Taenia coli are three bands of longitudinal smooth muscles in the muscularis of the large intestine. It would, the tension of the Taenia coli muscle as it pulls itself into a tone position would pull the walls of the intestine into large pockets like what you see now, which you call the haustra. So these are haustral pockets, what is caused by the tone of the Taenia coli. You also find small, like fat-filled pouches of visceral peritoneum, which you call the epiploic appendages. And up until now, no one can actually give us an idea of what these epiploic appendages is supposed to be for. It's not harming us though, but we never had a chance really to find out what those are actually for. So let's take a look at the subdivisions of your large intestine. The first one, the most proximal, which is connected to the ileum, is actually the sac-like pouch-like cecum. It's the first part of the large intestine. And together with that, there's actually a wall that's connected to the ileum and is guarded by the ileocecal valve. But right here on the most distal end at the base of the cecum is the appendix. The appendix is a mass of lymphoid tissue and is part of the mucosa associated lymphatic tissue of the immune system. It is a bacterial storehouse that is capable of recolonizing the gut whenever it's necessary. The twisted shape of the appendix makes it susceptible to blockages. Now after that, after the cecum comes the colon, the entire thing here that looks like a square wall that doesn't have any wall at the bottom. It's called the colon and it has several regions. The first region, almost all of the region is retroperitoneal, except for this region here on the top. This is actually like protruding in the front into the abdominal cavity, which is the transverse colon. So the regions are as follows. First, you have an ascending colon on the right side of, well, your cecum is actually on the lower right side of your abdominal pelvic cavity, and it is connected to the ascending colon. The ascending colon makes a right angle turn, and it forms a flexure, which is called the right hepatic flexure. It's called right hepatic flexure because that corner right there is where you will find the liver. On top of this area here is the liver. So after the right hepatic flexure comes the transverse colon, which is the only one that is intraperitoneal. If you're going to take a look at the entire large intestine, your your cecum, your ascending colon is actually retroperitoneal. Your transverse colon is intraperitoneal. Then your intraperitoneal sigmoid colon is now going to make a right-hand turn again and will form the splenic flexure. It's called splenic flexure because the spleen is right here on top of it. So after turning 90 degrees, it goes down and becomes a descending colon. The descending colon makes an S turn, which actually is the forming your sigmoid colon. Did I miss anything so far? So, in the sigmoid part right here, the intraperitoneal regions of the sigmoid part is anchored to the posterior abdominal wall by the mesentery sheaths, which you call the mesocolon. This one here is actually also the mesocolon, and here is actually the transverse mesocolon that holds the transverse colon. Now we're going to get into the anal canal, the last segment of the large intestine that opens to the body exterior at the anus. And it has two sphincters that is worth looking at. First, you have the internal anal sphincter. This is your internal anal sphincter. And that's another one. So this is a sphincter. That means it goes around the entire hole. And it's actually smooth muscle. And so being that it is a smooth muscle, it is involuntary. So this is actually receiving signals from the brain if you really need to go. If you really need to go, then it will relax and it will allow fecal matter to go out. But we have a way of controlling it, and our way of controlling it is with the use of the external anal sphincter, which is a skeletal muscle. So these are your three bands of internal anal sphincter, and all of them, one, two, three bands, all of them are skeletal in nature, and therefore you can control it by will. You can say no, you don't need to go yet because, or you don't want to go yet because you can't find a bathroom. So let's take a look at the mesenteries and their relation to the organs of the abdominal cavity so you will understand the rest of the other accessory coverings of the organs of the abdominal cavity. First of all, the yellow line, there you go, that yellow line right there is your boundaries to your abdominal cavities. All right. Now, on the border of the yellow line is a cover, the ink green. That particular cover covers the entire the wall of the abdominal cavity. And this is actually what you call the parietal peritoneum. So this green line is actually the parietal peritoneum. Now, the parietal peritoneum would have extensions of uh, what do you call this what you call peritoneal extensions, and these extensions would pretty much like go around organs slowly go around organs and then go out and connect and then go around organs again. And then sometimes they from the body wall they would pretty much like serve as an anchor of these organs to the body wall, and then they go around again and wrap themselves around the organ. When this peritoneum or peritoneal membrane wraps themselves around the organs, you call them visceral peritoneum. When they are covering the wall, you call them parietal peritoneum. When the intraperitoneal membrane would connect one organ to another organ, you call that a mesentery. So this is a form of mesentery, and you call this the lesser omentum. This is another form of mesentery. You call this the greater omentum. You you call this also this little um, thing that looks like spider, okay, that would anchor the intestines to the dorsal body wall. You call this the mesentery. Okay, so even the mesocolon is actually a mesentery. The mesentery connects an organ to another organ or connects the organ to the body wall. Well, why is it that this one here is called an omentum? Well, this one here is called an omentum. For instance, you call this the greater omentum because it's actually a mesentery that connects the lesser curvature of the stomach to the underside of the liver. Then this one here, the greater omentum, is actually called greater omentum because it's connected to the greater curvature of the stomach and is connected to the transverse mesocolon. Here you go, up front. Okay, in this view, you could see the lesser omentum and you have see the cut end of the greater omentum. And I think we have a model in the lab and in the pictures in your lab that would show pretty much something that's very close to this picture here. You will not see, there is no model that shows the lesser omentum, but you will see a model that shows the greater omentum on the greater curvature of the stomach. Here you go again, it's a good picture, okay? You have a good picture of the lesser curvature of the stomach and the greater curvature of the stomach. Lesser curvature of the stomach has the lesser omentum and it connects to the underside of your liver. The greater curvature of the stomach has your uh, greater omentum. And here, this is a mesentery. There you go. This is a mesocolon. The mesocolon is a mesentery of the large intestine. It's actually the one that uh, connects or hangs um, the large intestine to the dorsal body wall. It suspends them to the dorsal body wall. So this is a homeostatic imbalance. Here's a picture of an inflamed appendix, and inflammation of the appendix is actually appendicitis. Usually it results from a blockage by feces that traps infectious bacteria, and the most common, it's actually very common among children and adolescents whenever the entrance to the appendix is at its widest, which is actually because of their age. Venous drainage at this point can be impaired, leading to ischemia and necrosis, and therefore can result in oftentimes in ruptured appendix. Ruptured appendix, if it is not caught immediately, can cause peritonitis. Now, let's take a look at the microscopic anatomy. Large intestine. The large intestine contains a thicker mucosa, and it's made up also of simple columnar epithelium like your small intestine. except in the anal canal. The anal canal, it has high traffic, and therefore you don't expect it to be covered only with simple epithelium. The anal canal is actually made up of stratified squamous epithelium in order to withstand the abrasion or the abrasive nature of the movement of the fecal matter along the passageway. It does not contain circular folds or villi or digestive secretions. It contains abundant deep creeps with many mucus producing goblet cells. Most of the anal canal hangs in long ridges or folds referred to as the anal columns. Anal recesses is located between the anal columns, and uh, they secrete mucus to aid in emptying the pectinate line, the horizontal line that parallels the anal sinuses. Visceral sensory nerves innervate the area superior to this line. It is a region that is very sensitive to pain. Somatic nerves innervate inferior to this line. This region is sensitive to pain, and pretty much this is the reason why if we have inflammation of this particular area here, which actually what you call hemorrhoids, it is really painful. Let's take a quick look at the bacterial flora of your intestines. Um, the bacterial flora consists of about 1,000 different types of bacteria, more than 1,000 different types of bacteria. And these bacteria outnumber our own cells 10 to 1. They enter from the small intestine or anus to colonize the colon. And they do have metabolic functions. These bacteria help in fermentation. They ferment undigestible carbohydrates and mucin. They also release irritating acids and gases. About 500 ml of these gases is produced every day. But they're also responsible for a good amount of vitamin synthesis. They synthesize vitamin B complex and some vitamin K that is needed by the liver to produce the clotting factors. They are also keeping pathogenic bacteria in check. Beneficial bacteria outnumber and the pathogenic bacteria, and therefore they are able to suppress them. The immune system destroys any bacteria that try to breach the mucosal barrier. The epithelial cells recruit dendritic cells to the mucosa to sample microbial antigens and present them to our lymphocyte, to the T cells. That is actually, they actually like being carried, um, the lymphocytes actually are being carried by the blood into the mucosa associated lymphatics tissues of the gut, and this is where they're going to be meeting a lot of the pathogenic bacteria. So this would the presence of the T cells in the Peyer's patches, for instance, triggers the production of immunoglobulin A, and immunoglobulin A would be restrictive to the microbes. Now, there are mounting evidence that supports gut bacteria and health. Findings are that the kinds and proportions of gut bacteria can influence body weight, and also susceptibility to various diseases, including diabetes, atherosclerosis, and fatty liver disease. Our moods pretty much manipulate gut bacteria, uh, and therefore manipulating them in counter manipulating them may become a routine healthcare strategy in the future. Just imagine how many bacteria we have, more than 1,000. Okay, let's take a look at more homeostatic imbalance. Homeostatic imbalance associated with the gut is actually, one of them is antibiotic associated diarrhea, which accounts for about 14,000 deaths every year. And this is brought about by a bacteria which you call the Clostridium difficile. It is an anaerobic bacterium that carry many that is actually carried in the intestine. It is the most common. It's actually the most common source of the antibiotic associated diarrhea. Well, first of all, whenever you're taking a lot of antibiotics, when other bacteria are eventually wiped out by these antibiotics, then your Clostridium difficile can flourish without this helpful bacteria around, because they've been killed by too much antibiotic. And pretty much the presence of Clostridium difficile can cause us to develop pseudomembranous colitis, or inflammation of the colon. It may lead to bowel perforation and sepsis. So Clostridium difficile infections are resistant to many antibiotics and are difficult to treat. This is one of the craziest bacteria really to treat. So the new treatment includes fecal transplant, meaning transplant of feces, healthy feces, into the patient to replace the healthy bacteria, so that the Clostridium difficile can actually be can be killed by the pathogenic bacteria of the transplanted fecal matter, or fecal tissue. I don't know if I don't think they cut tissue from another person, but it's actually fecal matter from another person. So let's take a look at the digestive process in the large intestine. The residue remains in the large intestine for an additional 12 to 24 hours. So the undigested particles stay in the large intestine, is going to be processed there. There won't be any breakdown of food anymore, except what the enteric bacteria will digest. Vitamins, pretty much, and water and electrolytes are reclaimed. Major function of the large intestine here is actually the propulsion of feces towards the anus and defecation. The large intestine is important for our comfort, but is not essential for life. If the colon is removed, the terminal ileum can be brought out. So if you're going to be cutting the ileum right there before the ileocecal valve or whatever, I don't know how the surgeon would be doing that, then that particular terminal end can be brought out through the abdominal wall, and this procedure is what you call ileostomy. So in the end, the patient will need to be carrying a bag where that end of the ileum will be dumping all of the undigested material. Haustral contraction. These are your haustral pockets. Those are the pockets. The haustral contractions, most of the haustral contractions of most of the colon pretty much are sequentially happening in response to distension. If there's already too much food that's already accumulated there and they are getting distended, then they will start to contract. There's actually they start with slow segmenting movements, mostly in the ascending and the transverse colon. Now, once the gastrocolic reflex happens, which is actually initiated by the presence of food in the stomach, the gastrocolic reflex would result in mass movement. Even if you drank water only, most especially in the morning, you drank water, you had coffee, immediately you feel that you want to go to the bathroom and do a number two immediately, because you just initiated your gastrocolic reflex. And this resulted in mass movement. The mass movement is slow but very powerful peristaltic wave that is activated about 3 to 4 times per day. And this mass movement is capable of emptying half of the transverse colon all the way to the end of the sigmoid. All of the contents of that end goes out in that one shot right there. And then the rest of the other half immediately will be moved to replace that. And therefore, your peristaltic wave, your mass movement, can occur about 3 to 4 times per day, depending on how much food you've eaten that fills your colon. The descending colon and sigmoid colon act as storage reservoir, which is pretty much going to be emptied out in one mass movement, mass contraction. Let's go to the homeostatic imbalances. Now, low fiber diet can narrow colon and cause strong contractions that increase pressure on the walls. It can cause the formation of small herniations of the mucosa. They're like buckets, which you call the diverticula. And diverticulosis is a word we refer to when we say that there's actually diverticula in the walls. So a person will have a diverticulosis, and it's very common. This diverticulosis is common in a sigmoid colon. They affect half of the people already. That's actually like 70 years and older. Diverticulitis is your inflamed diverticula that may rupture and leak into the peritoneal cavity. So remember, guys, what I mentioned to you, and therefore right now you're still young, you still have a chance really to change your diet if you are not in your if you are into the diet that will cause the formation of diverticulitis. Low fiber diet, which is actually if you're not eating, if you're not fond of eating vegetables, then you might think twice because vegetable high fiber diet can actually normalize the size of your colon. Low fiber diet, which means if you eat a lot of meat and very little vegetable, you are going to be narrowing your colon and can cause strong contraction that could increase pressure on the walls that results in the formation of diverticula. And diverticulitis is actually the inflammation of this diverticula and may rupture and leak into the peritoneal cavity and may be threatening. And this is actually what you call the irritable bowel syndrome. It is a functional gastrointestinal tract disorder. It is the symptoms will be recurring abdominal pain, stool changes, bloating, flatulence, nausea, and depression. Stress really is one of the precipitating factor of diverticulitis. And therefore, stress management is an important part of the treatment. Then we go to defecation. So, mass movement forces the feces towards the rectum. There's actually distension, and that's actually initiated by a spinal defecation reflex, which is a parasympathetic signal. It stimulates the contraction of the sigmoid colon and the rectum. It relaxes the internal sphincter, and the internal sphincter is actually like you cannot control it, okay? And so when the brain says you relax and you let go of the contents of the large intestine, then it relaxes. But then there's conscious control allowing the relaxation of the external anal sphincter. And so whenever, if you don't want to, then you may control it. Muscles of the rectum will contract whenever you feel like you want to expel the feces, and is assisted by the Vasselva's maneuver whenever we're doing it, which is actually characterized by closing of the glottis, contraction of the diaphragm, and the abdominal wall muscles can cause increased intra-abdominal pressure. The levator ani muscle contracts, causing the anal canal to be lifted superiorly and allowing feces to leave the body. And that's how you do your number two. Aha. So here's actually another view of the defecation reflex. Okay, your feces is moved and extended into the rectum. Okay, let me just make that better. Okay, stimulating the stress receptors there, and the receptors transmit the signals along the afferent fibers to the spinal cord neuron, and that's actually how it is, and that's actually where it's coming from. The spinal reflex is initiated, which is a parasympathetic motor in which the parasympathetic motor fibers stimulate the contraction of the rectum and the sigmoid colon and the relaxation of the internal anal sphincter. And it is convenient. This is actually when, after that, the um, the internal anal sphincter is now relaxed. It is now convenient to defecate, and uh, the sympathetic neurons are inhibited. The parasympathetic system allows the external anal sphincter to relax so the feces may pass. Homeostatic imbalance. One of them, very common, is actually diarrhea. This is your watery stools that results in large intestine that does not have sufficient time to absorb or reabsorb the remaining water. Cause of this will be the causes include irritation of the colon by bacteria or by jostling of digestive viscera, and this occurs in among marathon runners. Prolonged diarrhea may result in dehydration and electrolyte imbalance. And if you have, and it will also result in acidosis and thus loss of potassium. Another one is constipation, which is the exact opposite of diarrhea. This can occur when food remains in the colon for an extended period of time and too much water is absorbed. The stool becomes hard and gets difficult to pass and may result from insufficient fiber. This may result from insufficient fiber, again, insufficient vegetable in your diet, or insufficient fluid in the diet, and improper bowel habits, lack of exercise, or laxative abuse. So, let's take, in a nutshell, let's take a look at digestion itself. In the digestion itself. Digestion starts from the mouth, okay? You're ingesting it, and pretty much these molecules are only when these molecules are small enough to be absorbed would they be able to cross the walls of the small intestine. Digestion, which is actually a chemical breakdown, is a catabolic process that breaks macromolecules down into monomers that are small enough for absorption. So not unless we're able to break it down into its smallest absorbable units would be able to really absorb them. The intrinsic and accessory gland enzymes are involved in the digestion of enzyme in the digestion process. And all enzymes are hydrolytic enzymes, where water is added to break the chemical bonds. Absorption is a process of moving substances from the lumen of the gut into the body. Okay, so here is a diagram showing us the mechanism of absorption. You have, first of all, you have tight junctions. Those are tight junctions right there. Okay, and that's another tight junction there. Tight junctions would make sure that the molecules pass through the epithelial cells rather than between them. So you're probably thinking the food goes right in between the cells. No, food goes right through the walls of the cells, passing right through the entire wall, the apical wall and the basal wall on on the base of the wall of the columnar cell. Materials would first enter through the lumen side, which is the apical membrane, and exit through the basal or base or lateral, basolateral membrane in order to reach the side where you find blood vessels. Lipid molecules can be absorbed passively. They can be absorbed passively through the membrane, but polar molecules must be absorbed by active transport. Most nutrients are absorbed before the chyme would reach the ileum. So, in other words, we can conclude that there is uh, not much absorption happening in the ileum anymore. Most of the absorption is happening in the duodenum and in the jejunum. So now let's take a look at how we absorb, how we process, which means how do we digest and then absorb all of the different nutrients that we have that's part of our diet. Meaning, how do we digest carbohydrates and absorb it? How do we digest proteins and absorb it? How do we digest lipase and absorb it? And how do we digest those vital DNA and RNA that we have, which is not really a carbohydrate, neither are they lipase, or rather, neither are they fats, and neither are they protein. They are nucleic acids. How do we digest the nucleic acids? Because they're part of the things that we are actually eating, because all of the things that we are eating are living, part of a living entity. Okay? So they do have nucleases or RNAs and DNAs, and we have to digest them so that eventually they become part of our cells and they eventually will become part of the DNA and RNA that we are producing, the human DNA and RNA. So let's take a look at uh, the first food stuff, which is carbohydrates. Here you have your starch and disaccharides. Let's take a look at this here now. And take note that only the monosaccharide, take a look at the bottom part. Only monosaccharide. These are monosaccharides examples: galactose, glucose, and fructose. Only monosaccharides can be absorbed. Starch and disaccharides are broken down into oligosaccharides and disaccharides, and then they need to be broken down as these monosaccharides before they get to be absorbed. Now, this digestion of carbohydrates begin in the mouth with salivary amylase, and it's broken down into lactose, maltose, and sucrose, which are disaccharides. And finally, these disaccharides are broken down into their monosaccharide units. For instance, your lactose is broken down, which is a disaccharide, is broken down into galactose and glucose. These are the two monosaccharides that form the lactose, right? Then your maltose is made up of two glucose molecules. It's a disaccharide made up of two glucose molecules and is broken down to two pieces of glucose. And your sucrose is actually made up of fructose and glucose. And when they are broken down, eventually they are broken down into their glucose and fructose units before they are absorbed. So we do not really absorb the lactose, the maltose, and the sucrose. They need to be broken down further into their smaller units. The brush border enzymes in the small intestines, which is actually the ones that would be acting on these disaccharides so that they are going to be broken down into their smallest forms, will be releasing dextranase, glucoamylase, lactase, maltase, and sucrase. So lactase is the one that is necessary in the breakdown of lactose into galactose and glucose. If you don't have lactase, if your body is not anymore producing lactase, then you become lactose intolerant. So what happens to this monosaccharides? They are now going to be absorbed via co-transport with sodium. First, in fructose passes via facilitated diffusion, and then all monosaccharides would leave the epithelial cells via facilitated diffusion. They would enter the capillary blood in the villi, and they are transported to the liver via the hepatic portal vein. Here is a closer view. You have here this is the lumen. This pink area is the lumen. And these four are actually the columnar cells of the gut. And that's your capillary right there. So your glucose, galactose, and fructose pretty much would be entering the cells and would be co-transported across the apical membrane of the absorptive epithelial cells, and they are mostly by secondary active transport with sodium. Monosaccharides exit across the basolateral membrane by facilitated diffusion. And right here gives us the steps of starch digestion in the intestine. The pancreatic amylase, right there, pancreatic amylase would break down starch or glycogen that escapes salivary amylase into oligosaccharides and disaccharides. Brush border enzymes, dextrinase, lactase, glucoamylase, maltase, and sucrase would further break these into lactose, maltose, sucrose, and then into monosaccharides, glucose, fructose, and galactose. I mentioned that already in a previous slide. Then the monosaccharides, like glucose and galactose, are transported across the apical membrane of the enterocytes. This active transport uses sodium concentration gradient established by the sodium and potassium ATPase pump in the basolateral membrane. And you have monosaccharides that exit across the basolateral membrane is transported by facilitated diffusion, and they would enter the capillary via the intercellular clefts. Let's take a look at the homeostatic imbalances involved in the large intestine, in the digestive process. Now, I mentioned people with lactose intolerance, they would be deficient in lactase and cannot consume lactose. And therefore, any lactose that remains undigested and creates an osmotic gradient in the intestine prevents water from being absorbed, resulting in diarrhea, and can also pull water from the intestinal space into the lumen. And bacterial metabolism of undigested solids produces a large amounts of gas, resulting in bloating, flatulence, and cramps. And some people, this reaction is actually like big. I'm also lactose intolerant, but none of this happens to me. I probably would have very mild bloating. That's it. That's the only thing that happens to me, but I know I am lactose intolerant. And therefore, the treatment will be adding lactase enzyme to milk or take lactase tablets before consuming milk products, or just consume lactose-free products. Now we go into the digestion of protein. Protein is very important because it is not that carbohydrate isn't, but right now we have a fad of how to lose weight by trimming down on our carbohydrates in the diet and eating more of protein. But then again, when we get into chapter 24, you'll find out that if you eat too much protein and you don't burn that protein, it gets converted into fat. So you have to be very careful with how you're eating your protein. Eating too much protein and overdoing it is also not good. Protein, or the source of protein, is not only dietary, but also includes digestive enzyme. All of our enzymes are made of protein. And proteins from also from the breakdown of the mucosal cells that is in our stomach is also going to be part of the diet. Not really diet because we did not eat it, but it's part of what is actually been part of the in the digestive process. So proteins are broken down first into large polypeptides. So you have your protein here broken down into large polypeptides and then small polypeptides. Finally, into amino acid monomers, which is actually with some dipeptides and tripeptides. And eventually, these amino acids are the ones that are absorbable. Digestion of protein begins in the stomach, right there. When pepsinogen is actually converted to pepsine at a pH of about 1.5 to 2.5. And that's the reason why hydrochloric acid needs to be produced. I'm not sure if you could see this there. I know you can see this. So if hydrochloric acid is present, then you are converting pepsinogen to pepsine, which is the active form. And this is the one that would digest the protein. This pepsine that is actually in the chyme of the stomach and that's continuously digesting protein. This will become inactive the moment the chyme in the stomach enters the duodenum because the duodenum, the pH in the duodenum is not anymore going to be 1.5 to 2.5. There's going to be like an influx of high alkaline mucus by the Brunner's gland and um, by the pancreatic juice that's going to neutralize the acidity of the chyme and bring it up to about 7. So, what happens to those digested items? Amino acids, for instance, in the stomach that are absorbed via co-transport with sodium ion. Some dipeptides and tripeptides are absorbed via co-transport with hydrogen ion and hydrolyzed into amino acids within the cells. In the small intestines, infrequently, there is transcytosis of small peptides that would occur. Amino acids would leave the epithelial cells by facilitated diffusion, excuse me, and they enter the capillary blood in the villi and are transported to the liver via the hepatic portal vein. Remember that every single thing that we absorb in the gut, whether it's the stomach or the small intestine, they all go to the bloodstream. But they're not just going to circulate, they need to pass by the liver so that the liver can process all of the food that we have taken. And the liver needs to make sure that whenever there are some poisonous molecules that we have absorbed, they need to be detoxified. So the liver has a big job here in screening the blood that is rich in nutrients that we have just eaten up for the day. And here is actually the steps in digestion of protein. Okay, first you have your pancreatic proteases that break down proteins and trypsin. These are your trypsin and chymotrypsin that cleaves protein into small peptides, while your carboxypeptidases takes off one amino acid at a time from the end. The brush border enzyme aminopeptidases, carboxypeptidases, and dipeptidases break oligopeptidases and dipeptidases into amino acids. So amino acids are then, once they're broken down and once you have already the amino acid, with the use of a carrier, they are co-transported across the apical membrane of the absorptive epithelial cells. This active transport uses the sodium concentration gradient established by the sodium-potassium ATPase pump in the basolateral membrane. Then amino acid exit across the basolateral membrane via facilitated diffusion and would enter the capillary via the intercellular clefts. That's cool. So again, let's look at another version of the different steps in lipid digestion in the intestine. And here we look at the emulsification. Here, this is actually where triglycerides are and their breakdown products that are insoluble in water. They need a pre-treatment. Right here, you could see the pre-treatment. They need a pre-treatment with bile salts. It's actually bile salts combining with the fat globules. So they break the large fat globule into smaller units. And digestion process start when the pancreatic lipases would break down fat into monoglyceride. And these are your monoglycerides. So here, digestion happens here. The pancreatic lipases hydrolyzes the triglyceride, yielding monoglycerides and 3 fatty acids. Then you have the formation of micelles. Micelles are substances that are made up of fatty acids, monoglycerides, and bile salts altogether. And they ferry their contents right through the epithelial cells. And this happens only by diffusion. Fatty acids and monoglycerides diffuse from the micelles into the epithelium. So now your micelles are left out, your bile salts are left outside. The micelles will be releasing only the monoglycerides that would diffuse into the epithelial cells and then into the lacteals. Here you're going to form chylomicrons. That's actually before it goes right through the basolateral membrane. There's going to be a conversion of the micelles into chylomicrons. Chylomicrons are fatty acids and monoglycerides that are recombined and packaged with other fatty substances and proteins to form these little nuggets of chylomicrons. The chylomicron is transported and extruded from the epithelial cells by exocytosis, and they would enter the lacteals and are carried away from the intestine through the lymph. They're going to go pass through the thoracic lymph, the thoracic duct of the lymphatic system, and eventually will be released together with the lymph into the subclavian veins. Then now you have the fat digestion here also. This is just you could actually just look at this chart, which is nothing but a repetition of what I already did. The emulsification of fat, digestion of fat by pancreatic lipases, gastric lipase, etc. Please make sure that you look into this and how they're actually absorbed. And finally, you have your nucleic acid, the DNA and the RNA of the living organisms that is part of our diet. So you digest the cells of the plant sources and animal sources of our diet. And you have the pancreatic ribonucleases and the deoxyribonucleases that breaks it down into it, and then of course also with the help of the brush border enzyme, which is the final breakdown. breaks down the nucleic acid into its original. Remember that a nucleotide is made up of the pentose sugar and your phosphate and the ion. And when you digest it, it's going to be a nucleotide is going to be broken down into its smallest unit, smaller than a nucleotide. Hence, those units would enter the intestinal cells by active transport via the membrane carriers. Units are absorbed into capillary blood in the villi and transported to the liver via the hepatic portal vein. Let's take a view at the absorption of vitamins, electrolytes, and water. Vitamin absorption happens in the small intestine. Fat-soluble vitamins A, D, E, K, ADEK, are carried by micelles and diffused in the absorptive cells like any other fat. Water-soluble vitamins, on the other hand, are absorbed by diffusion or by passive or active transporters. Vitamin B12, which is a large and charged molecule, binds with intrinsic factor. That's why you need intrinsic factor, and intrinsic factor is actually produced by the parietal cells of the stomach and is absorbed by endocytosis. That's vitamin B12. In the large intestine, vitamin K and the B complex from bacterial metabolism are absorbed. And here, electrolytes are absorbed, transported actively along the entire length of the small intestine. Iron and calcium are absorbed in the duodenum. Sodium absorption is coupled with active absorption of glucose and amino acid. Chloride is transported actively. Potassium diffuses in response to osmotic gradient, and it is lost if water absorption is poor. Usually the amount in the intestine is absorbed, and iron and calcium absorption is related to the body's needs. So ionic iron is stored in the mucosal cells with ferritin, and when needed, transported in the blood by transferrin. We do not really need much iron because we have it and we recycle the iron in our body. Only if we are, well, if we are females and we are still actively menstruating, we can lose a lot of whole blood, and that's how we lose iron, and that's how we have what you call iron deficiency anemia. Among women, it's very common. Calcium absorption is regulated by vitamin D and parathyroid hormone. So water. How is water absorbed? Well, about 9 liters of water, most from the gastrointestinal tract secretions, would enter the small intestine, 95% of which is absorbed in the small intestine by osmosis, and most of the rest is absorbed in the large intestine. The net osmosis occurs if concentration gradient is established by active transport of solutes. Water uptake is coupled with solute uptake. Now, malabsorption, or not absorbing nutrients, is caused by anything that interferes with the delivery of bile or pancreatic juice. For instance, a damaged intestinal mucosa, probably because of bacterial infection or some antibiotics, or maybe even chemotherapy. Okay. And another one is another type of malabsorption is gluten sensitive, is among gluten sensitive enteropathy. It's called celiac disease. This is common malabsorption disease. It's an immune reaction to gluten, which is found in wheat flour. Gluten causes immune cell damage to the intestinal wall and the brush border. The treatment will be to eliminate gluten from the diet. And so that pretty much like finishes our digestive system. I will see you in the next chapter. Ciao.